Greetings everyone. We are here in Wound-Isscr 2022 to present mesenchymal stem cells and wound release factors ameliorate healing in rat model of chronic wounds. These wounds are associated with obstruction and repair cascade, presenting a major healthcare burden with prevalence of 6.3%. The available treatment approaches are mostly management techniques. However, advanced treatment strategies are being explored, and stem cells are one of the most promising candidates in this regard. With their self-renewal, paracrine, and less immune modulation activity, they have shown to promptly heal a chronic wound. However, their functionality and localization is compromised at the site of chronic wound injury. The literature suggests conditioning of MSCs can potentiate the therapeutic effect of MSCs. So, considering this, in current approach, we isolated the factors released in the later phase of normal healing, conditioned MSCs with those factors, and transplanted these MSCs in the in vivo chronic rat wound models. Our study is divided into 3 phases. First, we isolated and characterized MSCs. In second phase, conditioned these MSCs with the proteins released in the later phase of normal and diabetic healing. And the last phase, transplanted these conditioned MSCs at the wound site. The MSCs were isolated from rat bone marrow, cultured and characterized via immunocytochemistry and flow cytometry. The cells containing positive markers of stem cells were then preconditioned with the proteins, and the cytotoxic analysis showed that the MSCs, when conditioned with the proteins released in the later phase of normal healing at a concentration of 10 microgram, were most viable, and this concentration and this protein was the least cytotoxic for the cells. Similarly, gene expression analysis also showed that the N9 treated MSCs have enhanced expression of proliferative and angiogenic genes as compared to the inflammatory genes. The scratch margin was also profoundly covered in N9 treated MSCs. Later, when these conditioned MSCs were transplanted in vivo model, the macroscopic analysis showed that the wound margin was efficiently healed by the conditioned MSCs. The expression of gene at the site of wound after transplantation of conditioned MSCs showed that the angiogenic and proliferative genes were more expressed in that area as compared to the other treatment groups. CD31 was also more expressed in diabetic wounds treated with conditioned MSCs, and the skin architecture was regained and functionality was restored in the treated group. So we conclude here that the conditioning of MSCs with proteins released in the later phase of normal healing potentiated the in vitro and vivo healing abilities of stem cells by stimulating downstream repair cascade, which aided unhindered progression of healing process and timely restored the integrity and functionality of the chronic wound.